looked around at everything living like giant trees or even just yourself in the mirror and wondered about the basics. Why does an elephant always have an elephant baby?
Right, always an elephant.
But then also, why are siblings, like, so similar but also so different?
That's the core of it, isn't it? That mix of consistency and variation. That's heredity. And people have noticed it forever, obviously. How traits get passed down.
Yeah, inheritance. Our sources mention humans noticing this way, way back, like 8,000 to 1,000 BC, using it even.
Exactly. Exploiting it for artificial selection, picking the best crops, the best animals. They knew that worked.
But they didn't know why it worked, not the mechanics of it.
Precisely. It was all observation, maybe some guesswork.
Yeah.
Which brings us to why we're doing this deep dive today.
Yeah.
We want to unpack the work of Gregor Mendel, mid-19th century.
Right. The monk with the pea plants.
The very same. He's the one who shifted thinking. He stopped seeing inheritance as just some kind of blended soup.
And started seeing it as like little particles.
Sort of, yes. Discrete units, things you could actually count and analyze statistically. That was the leap.
So this is really the beginning of modern genetics. How Mendel used logic and math with his garden peas to figure out the basic rules.
And he was smart about it. Very methodical. He picked 14 specific types of pea plants. Crucially, they were true breeding.
What does true breeding mean exactly?
It means they reliably produced offspring identical to themselves for a specific trait, generation after generation. Like a tall plant line that only ever produced tall plants when self-pollinated. Stable inheritance. It gave him a clean baseline.
Okay, a solid starting point. And he focused on specific differences, right? Not everything at once.
Yes, that was key. He looked at 7 pairs of contrasting traits. Made the problem much simpler.
So instead of like 50 shades of green.
Exactly. It was binary. Is the stem tall or dwarf? Is the flower violet or white? Is the seed round or wrinkled? Clear distinctions.
Makes sense. Much easier to track.
And using true breeding lines meant the parent generation, the pea generation, was genetically pure for that trait he was studying.
Got it. Okay, so let's get into his big experiment. The monohybrid cross. What does he do first?
He took 2 true breeding parents, showing one of those contrasting pairs. Let's use the classic example. A true breeding tall plant and a true breeding dwarf plant.
Okay, tall parent, dwarf parent, crosses them. What happens in that first generation of offspring? The F1 generation.
Well, this is where things got really interesting and challenged the old blending idea. If it was blending, you'd expect.
Medium height plants, kind of in between.
Right. But that's not what he saw. Not at all. The entire F1 generation, every single plant, was tall.
Whoa. All tall. So the dwarfness just disappeared completely.
It looked like it vanished, yes.
Mockery?
The dwarf characteristic was nowhere to be seen in that first generation.
Okay, but wait. If it just vanished, how did Mendel figure out it wasn't just, gone forever, destroyed somehow by the tallness?
That's the crucial insight. He theorized it wasn't destroyed, just overshadowed. This led him to the idea of dominance.
Dominance, meaning one trait sort of masks the other one.
Exactly. He proposed that these factors controlling traits came in pairs, and in the F1 plants, the factor for tallness was dominant, meaning it was expressed, while the factor for dwarfness was recessive, meaning it was present but hidden.
Silent.
Okay, hidden. So how did he prove it was still there, just lurking?
The next step. He took those F1 plants, remember they all looked tall, and he let them self-pollinate. Create a second generation, the F2.
And what happened then? Did the dwarf trait pop back up?
It did, and not just randomly. It reappeared in a really specific, predictable way. When he counted them up, and he counted thousands.
He was thorough.
Very. He found that about 3/4 of the F2 plants were tall, and 1/4 were dwarf.
3/4 tall, 1/4 dwarf. A 3-to-1 ratio.
A consistent 3-to-1 phenotypic ratio. The phenotype is the observable trait, what it looks like. Tall or dwarf. That 3.1 ratio, you see it pop up a lot in basic genetics examples. But observing it is one thing. Understanding why 3.1. That's the genius part, right?
Absolutely. That ratio was the clue. It told him something deeper was going on with the underlying instructions.
Mm-hmm.
To get that 3.1, you have to look beyond the phenotype, the appearance, and think about the genetic makeups, the genotype.
Okay, genotype. So those factors he talked about, that's what we now call genes, right?
Correct. Genes are the units of inheritance, and the different versions of a gene for the same trait, like the tall version and the dwarf version, those are alleles.
Genes are the instruction type. Alleles are the specific instructions, like height gene with tall allele and dwarf allele.
That's a good way to put it. And we use letters to represent them, usually a capital letter for the dominant allele, like T for tall, and and lowercase letter for the recessive allele, T for dwarf.
Okay, T for tall, T for dwarf. So genotype is the combination of alleles an organism has.
Exactly, and there are possibilities. An individual could have two identical alleles, like TT or TT. That's called homozygous.
Homozygous makes sense, like homo meaning same, TT or TT, pure tall or pure dwarf, genetically.
Right, or it could have two different alleles, TT. That's heterozygous.
Heterozygous, hetero meaning different, TT. Okay, but wait, if a plant is TT, what does it look like? What's its phenotype?
Back to dominance. Because T is dominant over T, the TT plant is phenotypically tall. It looks tall, even though it carries the allele for dwarfness.
Okay, so that's why the dwarf trait vanished in the F1 generation. Those F1 plants must have all been heterozygous TT. They got a T from the tall parent and a T from the dwarf parent.
Precisely. They all had the T genotype, so they all look tall.
Got it. Now let's connect this T date back to the F2 generation. The self-pollination of TT plants gave that 3.1 phenotypic ratio. What was the underlying genotypic ratio?
When you work out the combinations from self-pollinating a T plant, the math reveals a genotypic ratio of 1 TT, 2 TT, 1 TT, 1 TT.
1 TT, 2 TT, 1 TT. Okay, let's break that down. The TT plants are tall, the T plants are also tall.
Right, because T is dominant.
And the T plants are dwarf. So you have 1 plus 2 equal 3 parts tall and 1 part dwarf. Phenotypically, there's a 3.1 ratio.
Exactly. The 1.2.1 genotypic ratio explains the 3.1 phenotypic ratio. This showed inheritance wasn't blending, it was particulate. These factors, these alleles, they segregate and recombine according to rules, the rules of probability.
This is where it gets kind of mind-blowing. It stops being just about plants, becomes math. Pure probability.
It really does. This led to what we call Mendel's first law, the law of segregation. It basically says that when an organism makes gametes, sperm, or egg cells,
Sex cells, right.
yes, the two alleles for a trait separate. So each gamete only gets one of the two alleles. An F1 plant with T genotype makes gametes, and half get the T allele, half get the T allele.
That's like a 50-50 coin flip for each allele going into a gamete.
Exactly like a coin flip. And when fertilization happens when two gametes combine randomly, the probabilities of the resulting genotypes are predictable.
How predictable? Like mathematically?
Perfectly predictable using simple algebra. Think about the chances for the gametes from the self-pollinating T plant. There's a 12 chance of a T allele and a 12 chance of a T allele from both the male and female parts. The combinations follow the binomial expansion. 12T plus 12T squared.
12T plus 12T times 12T plus 12T. Okay, I remember this from school. You get.
You get 12T 12T plus 2 12T 12T plus 12T 12T, which simplifies to
1/4 TT plus 2 quarters, or 1/2 TT plus 1/4 TT.
precisely 14 TT, 12 TT, 14 TT. That's the 1.2.1 genotypic ratio derived purely from probability.
Wow, that connection between a biological process and a simple mathematical formula, that it must have been revolutionary. It made biology quantifiable.
Absolutely game-changing. It showed the process wasn't mysterious blending, but ordered recombination governed by chance.
Okay, so Mendel figures out the ratios and the probability, but doing that binomial expansion every time seems a bit cumbersome. Isn't there an easier way to visualize these processes?
There is, thankfully, though it came a bit later, developed by Reginald C. Punnett. It's the Punnett square.
The Punnett square. Everyone remembers those grids from biology class?
Exactly. It's just a simple graphical way to represent that same probability calculation. Makes it really easy to see all the possible outcomes of a cross.
How does it work again? You draw a grid.
Right. You put the possible gamete types from one parent along the top, and the possible gamete types from the other parent down the side.
Okay.
Then each box inside the grid represents a possible zygote, formed by combining the allele from the top and the allele from the side for that box.
Got it. So if we do our F1 self-cross example, TT crossed with TT, what does that look like?
You put T and T along the top and T down the side. Your four boxes inside would be top left, T from top, T from side, so TT.
Okay, TT in the first box.
Top right, T from top, T from side, so TT. Bottom left, T from top, T from side, so TT again.
2 TT's boxes.
And bottom right, T from top, T from side, so TT.
1 TT box. So the grid visually shows you 1 TT, 2 T, 1 TT, the 1.2.1 genotypic ratio.
Instantly. And from that, you can see the three tall TT and TT to one dwarf TOT phenotypic ratio. Just lays it all out clearly.
Super useful tool. But okay, let's go back to the real world, outside the Punnett square. There's still that problem you mentioned earlier because of dominance.
Yes. The practical challenge.
If I'm a pea farmer, or just Mendel in his garden, and I see a tall plant, I still don't know for sure if its phenotype is TT, homozygous dominant.
Or if it's TT, Heterozygous, because both look identical, both are tall.
Right. The phenotype doesn't tell me the whole genetic story. How do you figure out the genotype of a dominant-looking individual?
Mendel figured this out too with another clever experimental design, the test cross.
A test cross. Sounds like what it does. How does it work?
It's designed specifically to reveal the unknown genotype of an individual showing the dominant phenotype.
So how do you test our mystery tall plant?
The method is brilliant in its simplicity. You cross the organism with the unknown genotype, our tall plant, TT or TT, with an organism that you know is homozygous recessive.
So you cross the mystery tall plant with a dwarf plant, genotype TT.
Exactly. You cross it with TT, why the dwarf plant?
Because the dwarf plant can only pass on a T allele. It has no T to give.
Precisely. The homozygous recessive parent can only contribute the recessive allele T to all offspring. It acts like a clean background or a genetic window.
So whatever the offspring look like depends entirely on what allele they got from the mystery tall parent.
You got it. The recessive parent tests the genotype of the dominant parent. Let's think through the possible results.
Okay, possibility one. Our mystery tall plant was actually homozygous dominant TT. What happens when we cross TT with TT?
Right, the TT parent only makes gametes with T. The T parent only makes gametes with T. So all the offspring must have the genotype.
Phi.
Right. And what's the phenotype of T?
Tall. So if the mystery plant was TT, all the offspring from the test cross would be tall. A 100% tall ratio or 1.0 tall dwarf.
Correct. Now, possibility two. What if our mystery tall plant was heterozygous TT?
Okay, so T crossed with TT. The T parent makes gametes, half T and half T. The T parent only makes T gametes.
So what combinations do you get in the offspring?
You can get T from the mystery parent and T from the dwarf parent, making T offspring, which are tall.
Okay, half the offspring are TT tall. What's the other half?
You can get T from the mystery parent and T from the dwarf parent, making T offspring, which are dwarf.
Exactly. So if the mystery tall parent was heterozygous, TTT, the test cross offspring will be approximately half tall, TT, and half dwarf, TT, a 1.1 phenotypic ratio.
Wow. So the appearance of any dwarf offspring in a test cross instantly tells you the tall parent must have been carrying that hidden T allele. It must have been TT.
That's the power of the test cross. It uses the predictable ratios revealed by crossing with a homozygous recessive to uncover the hidden genotype. Takes the guesswork right out.
That's really elegant. You use something genetically simple, the recessive, to reveal something genetically hidden in the dominant form. It all comes back to those predictable mathematical ratios.
It does. So we've gone from just seeing traits passed down to Mendel meticulously counting P's, figuring out dominant segregation.
Uncovering the 1.2.1 and 3.1 ratios, linking it all to probability.
And developing tools like the Punnett square and the test cross to visualize and apply these rules. It shows that inheritance isn't random chaos. It's ordered and fundamentally predictable.
And thinking about this for you, the listener, this basic P plant model, these simple rules Mendel found, they are the absolute foundation for understanding all inheritance and variation. Human genetics, animal breeding, evolution. It all builds on this.
The reason you might have your dad's eyes but your mom's smile, or why some traits seem to skip generations, it's not magic. It's the shuffling and recombination of these discrete alleles following these predictable probabilistic rules.
It really changes how you see variation in the world. It's not just random difference, it's the outcome of a defined process.
And that predictability. It does make you think, doesn't it? If a simple test cross lets us know the hidden genetic makeup of a plant with mathematical certainty, well, the ability to predict traits based on parental genetics has profound implications.
Yeah, extending that beyond plants. What really strikes you the most when you think about Mendel's work? Is it the sheer simplicity of the 3.1 ratio hiding such complexity? Or is it the fact that life's blueprint follows these almost mathematical laws? What's the most surprising piece